I'm a clinical neurologist. I see patients in the region that have Parkinson's. This is one of the what we call last frontiers of biomedicine. Brain disorders in terms of therapeutics. There's very little that can be done for patients. And I've always been interested in understanding more about the mechanism. So I've been very interested in understanding how PINK1 kinase mutations lead to Parkinson's. So that's been the background behind the work.
P10-induced kinase 1, PINK1, is a serine-threonine protein kinase that possesses an N-terminal mitochondrial targeting sequence, a transmembrane domain, and 3 intersectional loops within its catalytic kinase domain. PINK1 codes for an enzyme that protects the brain from damage-mediated mitochondrial dysfunction and oxidative stress. The primary function of the mitochondria is to produce ATP, which provides cells with the energy required for essential functions. In functional healthy mitochondria, PINK1 binds the translocase of the outer membrane, TOM, which is a protein complex responsible for translocating proteins produced from nuclear DNA to the mitochondrial membrane for use in the oxidative phosphorylation. Following binding to the TOM complex, a rhomboid transmembrane protease, PARL, cleaves to PINK1, which is then degraded by an MG132 sensitive protease to maintain steady levels of PINK1. In dysfunctional mitochondria, after binding to the TOM complex, there is accumulation of PINK1, as it's not cleaved by PARL. The PINK1 becomes activated through dimerization and phosphorylation. It can also be indirectly activated through mitochondrial uncoupling. When activated, PINK1 phosphorylates a ligase called parkin and ubiquitin, which then stimulates parkin recruitment to the mitochondria. Once at the mitochondria, parkin attaches ubiquitin to substrates on the outer membrane, which triggers degradation of damaged mitochondria, e.g. mitophagy. The majority of mutations in PINK1 impair its kinase activity and prevent the initiation of mitophagy in dysfunctional mitochondria. A build-up of reactive oxygen species, ROS, and premature neuronal loss leads to autosomal recessive early-onset Parkinson's disease. Parkinson's is a neurodegenerative movement disorder caused by a loss of dopamine neurons in the substantia nigra region of the brain.
We showed that this drug, which had been previously discovered to be an anti-Helminth drug, because it was used to treat Helminth infections, could in vitro, in cellular assays, activate the PINK1 kinase, which is involved in Parkinson's. So the mechanism by which it's activating PINK1 is not direct, it's indirect. It's doing it through uncoupling the mitochondria. And so for that reason, I think that there is very little scope for this finding to be translated or advanced to a more preclinical or clinical project.
The anthalamic drug Niclosamide is a common treatment for tapeworms which acts through mitochondrial uncoupling. Mitochondrial uncoupling is the prevention of ATP synthesis in the process oxidative phosphorylation. Oxidative phosphorylation is a coupled reaction of the electron transport chain and ATP synthesis. The kinetic energy loss from hydrogen ions dispersing across the proton gradient produces heat energy and therefore repressing ATP synthesis while indirectly activating PINK1. This study found that with increasing concentrations of the niclosamide, there is an increase in PINK1 activation.